Okay, so good evening. Welcome to the live interaction session for week 7. Um, we are today discussing a few topics that were covered as part of this week, among which the major focus was around the phenomenon of aging, cell senescence, as well as the development of cancer. That is the neoplastic transformation of a normal cell into a cancerous or into a neoplastic cell, and of course the process of apoptosis, how it differs from necrosis, and so on and so forth. So in this session, we will be discussing some of these topics, not all of them, because a major portion has already been covered in the lecture. But we will be looking at some of the associated topics, some extra information, and we'll be looking at some sample questions for this week. And we'll also be discussing the assignment for the previous week, which is the usual process or the usual workflow that we follow through the live interaction sessions. Please keep in mind that apart from this session, we will have another session for week 8 in the upcoming week. And following that, there'll be one conclusive session, which will be to discuss any and every question about this course, about the topics that were covered in this course, anything associated to what has been discussed in the live interaction sessions, any extra information, any extra references that you might need, or anything that might be relevant to that might be helpful for the upcoming examination, and any other doubts or information that one might need. So with this in mind, we will be starting today's lecture. So first of all, I'll just share my screen. Okay, so first of all, I'll be starting off with a description of the topics that were part of the assignment for week 6. We had covered the organization of the genome, or organization of the chromatin, and how it is structured within the cell. What are the various levels of organization, how that is relevant, why it is important to have that organization, what is its physiological significance, what is the need to have such extensive organization, and how it is mediated and how it is controlled at the level of DNA, not just DNA, but also the proteins which are at the center, which are histones. What are the modifications that occur within these? And in an additional very interesting lecture, we had also understood the 3C assay developed by Pentaco's lab, and Professor Lalore as well as Professor Ganji had explained some extra rather advanced techniques as to how these organizations and how these modifications are being studied in today's time, and what are the updated technologies that have allowed us to understand more and more about these techniques and how they are helpful for us. So with this in mind, we will look at the questions that were part of assignment 6, and we'll see how we can troubleshoot such questions in the upcoming future if they are a part of the examination as well. So the first question is about the beads on string structure. When we refer to that, what do the beads exactly mean? So the beads are either A, nucleosome core particles, B, SMC protein complexes, C, DNA polymerases, or none of the above. So if you remember, this was the first level of organization that occurs with respect to the chromatin, where you have the entire stretch of DNA from which around 147 base pairs wrap around an octamer of histones, and they are connected by a stretch of unpacked or free DNA, which is the linker DNA, followed by another bead. So that resembles the beads on string structure. And in this beads on string structure, the beads will refer to the nucleosome core particles. That nucleosome core particle is made up of 147 bases of DNA, which is wrapped around the histone octamer. So that is what effectively refers to the nucleosome. If you remember, if you go back to your lecture notes from the previous live interaction session as well, you will see that there is another structure called as the chromatosome, which does not just include the core nucleosome, but also includes the histone H1, as well as some additional nucleotide bases to form the effective structure. And that chromatosome is a slightly bigger structure than the nucleosome because of the additional bases that are included. And at a stretch, all of these together form the beads on string structure, which we are familiar with, which is otherwise also called as the nucleosome fiber, which eventually leads to the formation of the 30 nanometer fiber, and so on and so forth. Higher and higher levels of organizations will then follow it up. The nucleosome core particle contains a histone, octamer, heptamer, nonamer, or decamer. Like I have just mentioned, of course, the core histone is made up of a histone octamer, which includes histone H2, H3, and H1 as on the outer ends. And we also have H2, H2B. Altogether, they form an octameric structure of a very tightly packed histone core around which the DNA can be wrapped. Now, this core is positively charged because of the enriched proportions of lysine and arginine within the histone tails, and they allow for proper interaction and, in fact, very strong interaction with the DNA which is wrapped around it. That obviously carries a negative charge because of the presence of the negative charges on the phosphodiester backbone. Altogether, this forms a very strong structure which has formed the basis of epigenetic modifications and chromatin organization in every living cell within a eukaryotic individual. So the answer to this question is, of course, octamer. Following this, the next question was about SIR2. SIR2 is a HAT, HDAC, PI3K, or none of the above. So HAT is histone acetyltransferase, HDAC is histone deacetylase, PI3K is PI3 kinase, or none of the above. So we had discussed the nature of one of the key modifications which occur and how that is responsible for controlling whether the chromatin would be relaxed or would be tightly packed, or how accessible that region of the chromatin would be to the transcriptional machinery and any other machinery which tries to read the information in DNA in that particular location. So for that to be taken into consideration, one of the key modifying enzymes is SIR2, which is a histone deacetylase. When we say deacetylase, it means it acts upon the histone and it is responsible for the removal of an acetyl group, thereby removing a negative charge and thereby strengthening the interaction with DNA by enriching the effective positive charge on histone, which allows it to closely bind or closely interact with the negatively charged DNA backbone, and therefore it heterochromatinizes that region further and makes it inaccessible to the transcriptional machinery or makes it transcriptionally silent. So, or repressed, let's not call it silenced. It can be accessed by the machinery if it really wants to, but that region is somewhat repressed as compared to when it is acetylated. So the answer in this case would be the HDAC. Then in question 4, we have been asked to pick the incorrect statement about SMC proteins, SMC or structural maintenance of chromosome proteins, which we also discussed in the previous lecture. We have been asked to pick the incorrect statement among these following. So the first is whether they are conserved, whether they can fold back onto themselves via anti-parallel coil-coil interactions. Two SMC proteins interact with each other at the hinge region, or none of the above. So if you see these properties, all of them are true. SMC proteins are very largely conserved across different organisms. They are folding back onto themselves. They are forming coil-coil interactions. And two proteins, they were sort of forming a beta clamp-like structure, which we had also discussed, which allows them to surround the DNA at the locus where it is binding. And therefore, all three statements are true. Therefore, none of these statements are incorrect. And so option 4 would be the answer. Then coming to question 5, TADs are, this is just a simple expansion of the abbreviation. So TADs are topologically associated domains. The other options are, of course, not true, where it has been asked whether they are transcriptionally activated domains or whether they are temporarily associated domains. No, they are topologically associated. Then in question 6, we are asked what is a histone fold. So if you remember, we had discussed, we had seen the comparative structure of each of the histones and what are the conserved areas in each and how they are oriented in a particular manner, which looks like a histone handshake because of the orientation of histones, which allows them to have the appropriate area for it, or appropriate surface area along which they are interacting, which allows for their proper and very compact packaging that is responsible for forming the octamer the way we know it. So the histone fold in particular was made up of 3 alpha helices, which were connected by 2 loops. If you remember, they were marked as alpha 1, alpha 2, and alpha 3. And this was conserved across all of the histones. Then coming to question 7, which of the following modifications are found in histones? Acetylation, methylation, phosphorylation, citrullination, all of the above or none of the above. So among these, we have discussed that histone acetylation, methylation, and phosphorylation takes place. We did not discuss citrullination previously, but if you read about it a little bit, it is simply the addition of a citrulline group, or to be more precise, it is the conversion of an arginine present in histones into citrulline. And what it effectively does is that because arginine by itself intrinsically is a positively charged amino acid, the conversion of arginine into citrulline reduces the effective positive charge on the histone octamer, and as a result of that, it opens up the so the way to think about it is that if we think of a basic threshold level, which is required, when we say threshold, we mean the minimal level or the default level of negative or positive charge which is present in this interaction. Whenever there is a fluctuation from it, or whenever there is a change, either a reduction or an increase in the negative or positive charges of DNA and histones respectively, that will affect the extent to which these two molecules can interact, and therefore that will effectively have consequences on the transcriptional accessibility of that region of the genome. So if we think of the default level of positive charge, which is present in histones because of its default composition, whenever the arginines that are present, and like we have discussed that they are, histone tails in particular are very rich in lysine as well as in arginine. So when these arginine positive charges are being neutralized because of the conversion into citrulline, the effective consequence of this is that the charge from the threshold level, the positive charge from the threshold level is reduced. And as a result, the interaction between the two, between DNA and between histones, is getting affected or is getting loosened, as a result of which it is believed that it relaxes the chromatin and it provides accessibility to the transcriptional and other machineries in that localized region where the conversion into citrulline has taken place. Therefore, citrullination of histones can be considered to be an activation mark because it is allowing transcription to proceed. So with this in mind, the correct answer would be all of the above, because acetylation, methylation, phosphorylation, and citrullination, all four of them can occur for histones, and they have transcriptional consequences in the long run. Then in question 8, to count the number of SMC24 condensing complexes at the loop neck using the double tether DNA assay, which fluorophore was used? So this was a question that is part of the one of the extra lectures conducted by professors. So in that particular assay, the particular fluorophore that was used was Atto 647N. The remaining fluorophores that you see here are very commonly used for any and every assay, for immunofluorescence assays in particular, just for labeling any particular protein within a cell. You can tag it with these fluorophores, which are GFP, RFP, and YFP, simply known as green, red, and yellow fluorescent proteins. And they're extensively used in biological research, and they're used for simply labeling or tracking the motion or localization of a particular protein within the cellular compartments. So in this particular case, the fluorophore that was used was Atto 647N, and that would be the correct option. And for question 9, we were asked that which of the following statements is not true. So sister chromatid cohesion promotes proper bipolar attachment by steric constraints and tension. Without condensation, lagging chromosomes can be cleaved off during cell division. The cohesin complex generates sister chromatid cohesion by tethering sister chromatids, all of the above or none of the above. So we have been asked which of the statements is not true. Among these statements, they all are correct, which means that indeed there is the involvement of proper cohesion for proper bi-orientation or bipolar attachment, and there is an involvement of steric constraints and tension in proper separation of the two sister chromatids in two directions. Secondly, if there is no condensation, then lagging chromosomes can be cleaved during cell division. The cohesin complex generates cohesion by tethering them. All of these options are true. And therefore, none of the above is the correct answer because among these statements, no, none of these statements are false. And finally, in question 10, this is a very direct question. The cohesin complex gets summoilated by which of the following? So summoilation was very briefly mentioned. And the protein or the complex which was responsible for the same is NSE1 or MMS21 that carries out the summoilation of the cohesin complex. So that would be the correct answer. So, with this in mind, we come to an end for the assignment questions for that week. And now we will look at some of the topics and some extra information regarding the topics that were part of week 7. So, among these, I hope it's visible. Yeah. So, as part of week 7, one of the preliminary topics that were covered were the ideas of senescence and aging. So, what is the meaning of cellular senescence? Cellular senescence is considered to be a stable and a terminal state of growth arrest. What do you mean by? You have to understand the implication of each of these terminologies. That it is a stable state. That means cells are entering this state and they are remaining in this state for a definite period of time. It is a terminal state of growth arrest. It means that once cells have entered senescence, they will not backtrack. They will remain in that state. They will remain in that arrested state and will not, ideally, not enter into a state of proliferation again. Now, in this state, according to the definition, it is being said that cells are unable to proliferate despite optimal growth conditions and mitogenic stimuli. So that is what will distinguish it from the processes of apoptosis or autophagy, where the cell is under a very stressful condition, and therefore the ideal option for it is to undergo, is to die, simply to die. And therefore, despite optimal conditions and despite availability of nutrients, availability of mitogens, when a cell is deciding that it does not, or when a cell is preferring not to go into a state of division and is preferring instead to stay in this state of terminal growth arrest, then in such circumstances, we say that the cell has entered a state of senescence. So senescence is considered to be a hallmark of aging, which is something I'll come to in the next slide. But what we are looking at here is that there are a lot of factors which affect senescence. I think it was one of the preliminary diagrams that was presented in the lecture as well. And for your own reference, you can go to this review. It's a wonderful review with a very, very wholesome description, very comprehensive idea of what is senescence, what are its implications, and also it will give you an idea of what exactly is the current updated status about senescence, how research is being carried out in this field, and what are the directions in which you can look at research if that is something of interest to you. So I've mentioned it below. It was published in Nature Reviews Molecular Cell Biology, and it is a beautiful description of this entire topic of senescence. So what we see here are that there are different aspects with respect to senescence that have been looked at, mainly three. We have DDR or DNA damage repair, we have telomere shortening and damage, and we have oncogene activation, all of which are eventually amounting to senescence features. And we are seeing different types of changes that occur, including morphological changes, prolonged cell cycle arrest, oxidative damage, higher apoptotic resistance, SASP, which was also talked about, and of course, an increase in SA beta gal. So, let me just read out the legend once for your own understanding. So first aspect that we look at is that nuclear DNA damage is often causatively associated with senescence establishment. So one of the major causes that is associated with the with proceeding for senescence, when a cell chooses to go into senescence or considers it appropriate to go into senescence, is when there is extensive nuclear DNA damage. So DNA damage, what it does is that it activates a signaling cascade, which is identified as DDR or DNA damage response. And that is very commonly classified or commonly detected or characterized by phosphorylated histone H2AX or gamma H2AX. If you remember, this was something we also discussed in the previous live interaction session, where among a number of different histone variants, we saw that under certain conditions, under certain crisis, the cell chooses to replace its canonical histones with some sort of variants. One of them is gamma H2X, where there is phosphorylation of gamma H2X can very commonly be identified or can very directly be observed under the microscope. So when there is a stressful condition where a cell is entering into this state of, or when the cell has incorporated extensive DNA damage repair, then there are you can observe them as little dots, otherwise understood as foci, foci, which are little clusters of gamma H2AX. And phosphorylated gamma H2X can be observed in this regard, which is understood as locations where the repair machinery or the DNA damage response machinery has assimilated or has accumulated in order to carry out repair. So that is what is understood as a cell or gamma H2X foci or gamma H2X puncta are usually identified as a marker for extensive DNA damage. So coming back to the legend here, we are seeing that phosphorylated gamma H2X, 53BP1 and MDC1 are the typical, the apical kinases, ataxia, telangiectasia, mutated ATM and ATR, and the downstream kinases check1 and check2. So what effectively happens is that when there is a DNA damage, there is the assembly of gamma H2X along with MDC1 and 53BP1. And the presence of these or the presence of DNA damage overall is sensed by two sensor kinases, which are ATM and ATR. They are named so because they were identified from ataxia telangiectasia mutants or individuals who suffer from this particular disease. And these two sensor kinases undergo phosphorylation, indicating that there is a DNA damage or there, within the environment, there is some DNA damage that has occurred within the cells. After ATM ATR kinases have sensed the presence of a damage, they phosphorylated the downstream kinases check1 and check2. These are checkpoint kinases, which if you remember is something we had discussed even during the initial cell cycle classes. So check1 and check2 phosphorylation is responsible for phosphorylating P53, and it pushes cells towards the cell cycle arrest. So when this is one of the ways, or it controls P53, as a result of which it does not allow cell cycle to proceed through. So signals ultimately converge on P53 activation, which in turn elicits cell cycle arrest. When there is prolonged DDR activation, it triggers senescence. So when for an extended period of time, cells are in a condition where they are sensing extensive DNA damage, then they can enter a state of senescence instead of further proliferation. One or a few DDR signaling telomeres, the ends of chromosomes, are sufficient to trigger replicative cell senescence. So when there is extensive DNA damage towards the telomere, then under such circumstances, it is capable of pushing the cells towards cell cycle arrest and entering into senescence. Oncogene activation, the third arm, is also a powerful senescence trigger. Specifically, most activated oncogenes, partly via reactive oxygen species or ROS production, induce hyperproliferation and alter DNA replication patterns, which ultimately result in replication stress and DNA damage accumulation at fragile sites, which ultimately which includes telomeres as well. Besides prolonged DDR activation, senescence features also include cell cycle arrest by upregulating P21 and P16 inhibitors, which is what you see in the diagram as well. Oxidative damage, which is detected by increased ROS, the upregulation of BCL2 family of anti-apoptotic proteins, which induce resistance to apoptosis, metabolic changes, including senescence-associated beta-galactosidase or SA-beta-gal accumulation, which is what you see in the figure on the right as well. What you see on the left is a normal cell versus on the right, you see cells, a population of cells that have entered senescence, and it is it's a staining or colorimetric assay to detect for cells that have very high or that have upregulated beta-galactosidase activity, beta-galactosidase activity, and you can see that in senescent cells, there is significant upregulation and accumulation of this particular enzyme, and that is the easiest marker to detect the same. And apart from that, we also have SAHF, which is senescence-associated heterochromatin foci, which you can see inside the nucleus, and also SASP, which is a form of response, senescence-associated secretory phenotype, or SASP response. So all of these are characteristic features of the process of senescence, and altogether they have been um, understood and have been considered to be as hallmarks of aging. They're part of aging, but senescence in particular is the result of a number of processes. So, what you see on the left are the hallmarks of senescence, and senescence by itself is a central hallmark also of aging. So, what are the factors which contribute to senescence? We have epigenetic factors which can contribute to it. We have telomere erosion that we just talked about. That if there is, when we say erosion, it means that chunks of the telomere ends have been lost. They are no longer present, and therefore, whatever information is present on that particular chromosome cannot be relied upon or is variable and is not trustworthy in the long run. Then we have DNA damage, which we just talked about, that when the DDR or the DNA damage response is kept activated for a very long time, for an extended period of time, then cells also enter into senescence. When there is mitochondrial dysfunction, that if a cell has erroneous mitochondria which are not properly functioning, it is affecting the growth and in fact affecting it is affecting the production of energy. Not just that, it can also abruptly lead to some disruption in the mitochondrial outer membrane or the membrane architecture that leads to the anomalous release of some apoptotic or anti-apoptotic factors. Then that can also have a number of problems. In fact, if you have read about it, there are a number of mitochondrial diseases as well, which are extremely severe. Most of them are quite lethal to individuals, or they have very they severely affect the quality of life of individuals who suffer from the same. And if they are genetic in nature, you will see that there is a pattern of inheritance as well. Mitochondrial disorders usually have a maternal inheritance, and why that happens is something you will very easily find out. So if that is something you which interests you, you can look it up. But it is basically because of the way gametes are formed. That is the hint that I will give. And for your interest, you can go back to it and find it out. And if it is something that you're not able to clearly figure out, we can have a discussion on this in the last interaction session on 21st. So, yeah, these are the common hallmarks. And also we have stem cell exhaustion as well as chronic inflammation, which are the results of senescence, not the contributors to it, but they are a consequence of cells that are entering senescence. And we see that proteostatic dysfunction as well as nutrient signaling dysfunction have sort of a crosstalk with senescence. They can both be the cause as well as the effect of senescence, and therefore the indication has been shown as double-headed arrows. So again, this was part of the reviews, one of the oldest reviews that discusses the topic of senescence as well as aging, and you can look it up for your understanding and for your interest if that's something that you wish to pursue in the future as well. Now, uh, with this in mind, we are going to shift gears and we are going to look at two other more slightly different but very equally interesting topics, which are very, very commonly observed in living cells, but living cells that are encountering a slightly difficult situation. When we say slightly difficult, we mean that here cells are choosing to die despite the option of going into senescence. Cells are choosing to die because that is more appropriate based on the sort of condition that it is in at that point of time. Or it could be because it is part of the developmental program and therefore its death is required for the proper development of the embryo and proper development of the individual, their limbs or some sort of a structure to properly develop. So that process, which I am vaguely mentioning, is apoptosis. So the histological section that you see on the upper right is of apoptotic cells. So what you can see is in a tissue, while most of the cells are arranged in this sort of mesh-like manner, there are certain chunks in between or regions in between where we see that they're surrounded by a gap. So these cells are shrunken cells within which the nucleus is sort of collapsed within itself, or it has sort of condensed within itself, and the cell has shrunk. It has separated from the rest of the environment, and therefore they are undergoing apoptosis and they are dying in a very disciplined manner because that is the condition that it is in. In contrast, the histological section that you see on the bottom right is of a different section. It is of a section where we can see that the center of the cell is completely it is scattered, and it is so it looks like cells there have sort of burst and they have undergone a process which is not very properly regulated and not very well coordinated, and therefore these cells are said to be undergoing necrosis. Now, both of these processes are physiologically relevant. It depends upon the context in which it is occurring, the need for the process, and what exactly are the environmental conditions which are either supporting or opposing the same. So what you see here is it's a section that shows that there's a central mass or a central core where necrosis is taking place, and it is surrounded by a population of normal cells. All of which are together coordinating this entire process. And this is just one example of the types of the many types of necrosis that can occur. That is gaseous necrosis. We have liquefactive, we have some concentric, etc. There are different types of necrosis. Now, that is a separate process and a rather complicated process, so we'll not get into it. If it is something that interests you, you should certainly go back and check that out and see what information you can get in this regard. Now, with this in mind, we will um, we will now try to look at the differences that persist between these two processes, which we are saying are both physiologically relevant, but how are they different and what is the need for having these two visually different types of processes in place when effectively they are both causing cell death. So apoptosis, like I have mentioned multiple times, is the process of programmed cell death. When we say programmed cell death or PCD, it basically means a process which is being coordinated and regulated by sets of proteins, and therefore the cell is deciding to carry it out in a very organized and very disciplined manner without harming the surrounding cells or without compromising the health or survival of any other surrounding cell. In contrast, necrosis is often described, while it is not completely accurate, necrosis can often be described as premature cell death because it is not being carried out in a regulated manner. It is not always necessary, but it is occurring because of the type of conditions that it is facing. Apoptosis occurs through the shrinking of the cytoplasm followed by condensation of the nucleus. The histological section that we just saw also validates exactly that thing, that the cell cytoplasm has sort of shrunk, as a result of which the cell is much smaller, and the nucleus or the nuclear material that was there has condensed and has become a more darkly stained or more compact region within which all the genetic information is being assimilated. In contrast, when we look at necrosis, it has a completely opposite morphological um, manifestation. So it occurs by swelling of the cytoplasm. So the cell, instead of shrinking, it is swelling up. It is becoming much bigger. And it also has swollen mitochondria followed by ultimately cell lysis. When we say lysis, it means the cell ruptures or the cell bursts and all its interior contents are being released into the surrounding environment, which is why that process is considered to be not a form of programmed cell death directly, or not a very organized process, and something which can have an effect upon cells in the surrounding environment and can affect their survival or their action or their proper sustenance. So, apoptosis, in the third point, apoptosis is a naturally occurring physiological process. Necrosis, on the other hand, while I said that it can occur physiologically, it is not the preferred route. It is not something that the cell needs, but it does occur within a living system. Now, when does it occur? It occurs because of some external stimulus. When I say external stimulus, it can be by a byproduct of the presence of certain agents like toxins, trauma, some sort of a accident that might have happened, which can lead to necrosis. Very commonly, what you'll find is that in many patients who have undergone undergone some sort of a surgery after, not even undergone surgery, right after their accident, you can often see that there are reports. If you look at any of these medical shows, you will see that their limb starts to die. For example, if somebody has, you know, had an accident where they have injured their leg, then you can see in many cases that the tissue, it starts to change color, it becomes very severely inflamed, and that is when necrosis can occur. In a lot of cases, tattoos, people who get tattoos are advised to get them from very hygienic places, very trustworthy places, because if the needle is something that is not properly cleaned or that is not safe, it can actually cause inflammation because of some sort of a bacterial infection or any sort of infection, because it's piercing directly into your skin. In such cases, also it can lead to severe inflammation and can often cause the death of the muscle cells in that region, and therefore that such a limb will certainly not be useful, and many a times it can lead to an amputation or very severe cases. So necrosis occurs under such dire circumstances normally, but apoptosis is a more regulated, more controlled, and more micromanaged process inside a cell. The chromatin is aggregated during apoptosis, but in case of necrosis, we do not see any structural changes. Apoptosis is a caspase dependent pathway, whereas necrosis is a caspase independent pathway. Apoptosis is a localized process. So yeah, we're talking about not getting into much detail about caspases here, because caspases, if you've looked at the lecture, they are basically cysteine aspartases. That is where the name comes from. C for cysteine, ASP for aspartate. So these are cysteine aspartases because they have a cysteine in their active site. They're dependent upon the presence of cysteine for their activity, but their cleavage activity occurs after the aspartic acid residue, therefore giving rise to their name. And they are the most critical mediators of the entire process of apoptosis, which is a caspase-dependent pathway, whereas necrosis, of course, does not take the same route, and it is an independent pathway. Next, we see that apoptosis is a localized process. It is responsible only for the destruction of individual cells, whereas necrosis can affect an entire population. Like I said, that the process of necrosis is not very well controlled, not very well regulated, and the effect that it has because of the cell lysis or the cell rupture that is occurring, it can have the impact upon the surrounding cells as well, and therefore the effect can manifest in a group of cells instead of just the individual cell that is going through this process, and therefore it makes the entire phenomenon quite poorly controlled. Um, Apoptotic cells can be phagocytosed either by phagocytes or by adjacent cells. Um, yeah, even necrotic cells will be phagocytosed by phagocytes, because of course the cell debris that has been generated will not persist, because that is not healthy. That has to be cleared off because it can otherwise have poor consequences on the surrounding cells, and it will also remain as unwanted material or toxic material inside the body, and therefore its clearance has to be carried out, and that is taken care of by the phagocytes. Apoptosis, while it is mostly beneficial, there are cases where its abnormal activity can cause diseases. Even in the lectures, you had seen that its abnormal activation can its inactivation is often associated with cancer, but um, even the oncogenic activation of apoptosis has been recognized, and that can also happen. And that is what we see in case of apoptosis, or when it is reduced. And in fact, when there is too much apoptosis, that can also cause a number of diseases. So a little or too much of anything is, of course, not good enough, and it is associated with a group of disorders, not just one. You can look at reviews for this reference as well. There are a number of neurodegenerative disorders, number of cancers, and apoptosis has a differential role and a differential regulation involved in each case, and therefore the consequences that it has are also different. We will be looking at it a little bit in the upcoming few 2-3 slides, and maybe that will give you a little more clarity in this regard. On the other hand, when we talk about necrosis, it is harmful to the organism, certainly harmful to the cell, and certainly harmful to the organism as well at large. But despite this, it does occur. It is not desirable, but it does occur in case of necrosis. Pre-lytic DNA fragmentation occur, and here post-lytic DNA digestion occurs. So it means that before lysis, in case of apoptosis, so cell that will undergo apoptosis first carries out the fragmentation of its DNA and then undergoes the lytic steps. Whereas in case of necrosis, after the cell has lysed and all its digestive enzymes have been released into the surrounding cytoplasm, only then can the genetic material within be digested. And it is involved, apoptosis is involved in controlling the cell number within the body, because it has a, like I said, it has a developmental role to play as well in order to control the way in which an organism will develop. For example, one of the common understandings is in fact your hand, right? So during development, we are hands and our feet are said to have webbing between them, because the digits, they are all linked to each other by flaps of tissues. And this tissue, the reason why we have clear digits now, or we have separate digits now, is because the flaps, or the thin layer of tissue that is present between these digits is usually digested, and that digestion leads to the formation of the limbs and digits as we know today. So that is desirable apoptosis, and that is necessary for controlling the cell number within the body as well. On the other hand, necrosis is involved in the induction of the immune system and in defending the body from the pathogen. So it's part of the immune surveillance mechanism and part of the execution of the same process as well. So with this in mind, we will now go into details about the process of apoptosis. So apoptosis basically is, like we said, has a number of properties, but its regulation is mediated by, of course, caspases, which are the most key mediators of this pathway. But not just that, they are also very significantly affected by an entire family of genes, which are known as the BCL2 gene family. Now, this BCL2 family has two sets of proteins. We have anti-apoptotic members and we have pro-apoptotic members. Anti-apoptotic members are those that are opposing the process of apoptosis, and they are pro-survival. So we have BCL2, BCLXL, BCLW, BF1, BRAG1, MCL1, A1. So this is a snapshot from a relatively old paper. I think it's from 1995, if I'm not wrong. And because this is such an old paper where the initial, like the landmark studies on apoptosis had been carried out, the classification has been done differently. You will find a different subclass now of BH3 only proteins as well, which is something that you will find in the lecture notes also. So these are the anti-apoptotic members, and we have pro-apoptotic members, which are pro-death or pro-apoptosis, which are supporting apoptosis, and that includes BAX, BAC, BCLXS, BAD, BID, BIC, as well as HRK. So there is an entire family of proteins broadly classified into anti-apoptotic, pro-apoptotic, and BH3 only proteins, which are all together mediating this entire phenomenon of apoptosis within the cell in a very programmed manner. So keeping this in mind, we will now go into, first of all, the basic process of apoptosis. So again, the review paper that has been cited here is an excellent paper for understanding what apoptosis is, how it is being regulated, and it gives you a very comprehensive view of the entire topic. So all of the images that you'll find, or all the schematic diagrams that you'll find in this presentation particularly, have been taken from that particular review, and it shows the crosstalk and the interconnection between all of these pathways in a very beautiful manner. So if you can, if you have the time, and if it is something that is at the level of your understanding, if it is something you are able to do yourself, then please go ahead and look into this review paper. If required, we can also discuss it in details later on. So the apoptotic pathway, like I've already understood in the lectures, is made up of two or consists of two major branches or two major groups, the extrinsic and the intrinsic pathway. So as the name indicates, the extrinsic pathway involves some external factor, some external stimulus, which is a death signal or some sort of a anti-survival signal. Let's think of it in that way. And the intrinsic pathway, as the name indicates, involves the initiation of apoptosis from within the cell, that there is some sort of an intrinsic stimulus that is leading to the program or the initiation of a program that is triggering cell death. So in the extrinsic apoptotic pathway, I'm reading out the legend right now. Upon binding to the cognate ligand, we have death receptors such as TNF or tumor necrosis factor related apoptosis inducing signals. So you either have TNF or you have trail, both of which are being shown here, and you have FAS. All of these are different types of receptors. These are just names of receptors, and these are cell surface receptors. Very naturally, they have to be present on the surface because the signal is coming from the outside or is an external stimulus that is coming in, and then it binds to the cell surface signal and it initiates a downstream cascade. So these extrinsic signals, they come and bind to, or these extrinsic ligands, they come and bind to their cell surface receptors, cell surface depth receptors, and upon their binding, they activate initiator caspases. Here it has been shown in blue. We have procaspase 8 and procaspase 10, and this pro basically means that there is an additional domain which is maintaining that caspase in an inactive manner within the cell under normal circumstances, or under physiological conditions, or to maintain homeostasis basically. When there is this induction of death signals, or the involvement of death signals which are triggering the entire process of apoptosis, only then will the procaspase be activated. This pro domain would be cleaved on, and they would form the active versions of these enzymes, caspase 8 and caspase 10. And this is mediated because of the dimerization of the adaptive proteins, which is mediated by FADD or fast-associated dead domain proteins. So you can see the schematic representation on the left as well, that there's a death receptor to which the ligand is binding, and the downstream FADD, which is formed or which is associated because of dimerization of the death receptors, is activating the procaspases into active caspases. Now, active caspase 8 and 10, shown in blue, are then responsible for cleaving and activating the effector caspases, caspase 3 and caspase 7. So these initial caspases, that are the preliminary sensors of this death signal, are said to be initiator caspases, and what are actually executing the activity or executing the activation of apoptosis inside the cell, they are known as effector caspases, caspase 3 and caspase 7. This leads to apoptosis. This is the extrinsic pathway that we talked about. Alternatively, the intrinsic, otherwise known as the mitochondrial pathway of apoptosis, that requires the MOMP. What it means is that the mitochondrial membrane, if you remember the structure of mitochondria, it is a bilayer or a two-layer a double membranous structure that is present inside the cell. So you have an outer membrane, inner membrane, the intermembrane space, and within the intermembranes, within the inner mitochondrial membrane, you have the stroma, within which the cristae of the mitochondria are arranged, where oxidative phosphorylation takes place. So when there is permeabilization of the outer membrane of the mitochondria, outermost layer of the mitochondria, that is defined or described as MOMP, which is mitochondrial outer membrane permeabilization. So cell stresses, whenever there is a stressful condition under which a cell is deciding that it needs to die, what they do is that they engage BCL2 homology domain 3, or the BH3 only group of proteins that I talked about. That leads to the activation of BAX and BAC, and that triggers this mitochondrial oxidative phosphorylation. So what you see here, even in the schematic, you are seeing that there is this involvement of intrinsic lethal stimuli such as DNA damage, ER stress, hypoxia, and metabolic stress, all of which are effectively leading to the activation of BAX and BAC. Even BID, which leads to formation of truncated BID, is doing a similar thing, and it is activating these two proteins. What it does is that it causes the permeabilization of the outer mitochondrial membrane and the release of cytochrome C. Now, cytochrome C is the key signaling protein involved in the extrinsic pathway. If you have studied about secondary messengers inside a cell, mostly calcium is believed to be a secondary messenger, or cyclic AMP is believed to be a secondary messenger. What it means, secondary messenger basically means that These are molecules or ions or proteins inside the cell that are conveying some sort of information from one part of the cell to the another to another. They are not doing it directly because they are not engaged with any direct change in the cell surrounding, but the action of some primary messenger with the outside of the cell or with some intrinsic signal is changing the level of the secondary messengers. Similar to that analogy, cytochrome C um, is the key signaling molecule or is the key messenger in case of apoptosis, that is controlling whether a cell will enter apoptosis or not, and it can flip the balance in either direction. So the release of cytochrome C is a key event that takes place here. Following MOMP, I'm reading the legend again. That is, after the outer membrane has been permeabilized, the mitochondrial intermembrane space proteins such as SMAC, which is second mitochondrial derived activator of caspases, which you see here in pink, and cytochrome C, both of these are released into the cytosol. This cytochrome C can interact with APAF1, which is apoptotic protease activating factor 1, and it triggers the formation of this micelle-like structure. It's not a micelle, but the structure is analogous to that of micelles. It triggers the formation of the apoptosome, which activates caspase 9. This activated caspase 9 is then activating caspase 3 and 7. So we are again coming back to, or we are merging both of these pathways at this point, that both extrinsic and intrinsic pathway are dependent upon the same effector caspases, that is caspase 3 and caspase 7. Just the way in which this signal is being relayed is different in case of extrinsic and intrinsic pathways. So it eventually leads to apoptosis. The mitochondrial release of SMAC, which we talked about just a little while ago, it facilitates apoptosis by blocking the inhibitor of caspase, which is an X-linked inhibitor of apoptosis protein, or XIAP. So it is shown here in blue. So what it does is it blocks the blocks XIAP and caspase 8 cleavage of BID, which is a BH3 interacting death domain agonist, BID protein, enables the crosstalk between these two pathways and therefore mediates the funneling or the alignment of both the intrinsic and extrinsic pathways and controlling whether or not a cell will enter or not enter apoptosis. So this is the sort of point of merging of both of these pathways, and they are crucial for not just mediating apoptosis, but controlling cell survival and cell sustenance as well. So this was the default pathway that we talked about. Now, what we are now interested in is the fact that there can be, while the concept of apoptosis is something that we usually understand to be beneficial within a cell, that when a cell is encountering some sort of a stress or when it is not healthy, it should die, and therefore apoptotic mechanisms must operate in such cases, and that is the default route which is taken. There are exceptions to this. In fact, there can be pro-oncogenic effects. Oncogenic effects simply means something that will allow a cell to become oncogenic, to become neoplastic or become cancerous. So there can be pro-oncogenic effects of apoptotic cell death as well. How is that happening? So you will read the legend and look at the schema simultaneously to get an idea about it. What you see in the first part of this schematic, in part A, is a tumor cell which is currently dying. So during apoptosis, caspases, they cleave and activate one particular protein or one particular enzyme, calcium independent phospholipase A2 or IPLA2, which leads to the generation of prostaglandin, which is PGE2. So what you see is within a tumor cell, which is undergoing apoptosis or apoptotic cell death, you are seeing that the active caspases, apart from doing their destined job, they are also leading to the activation of one enzyme, that is IPLA2. What does it do? It releases arachidonic acid, which activates COX-1 or COX-2, which effectively leads to the release of prostaglandin E2. That is the net molecule that we can see is produced as a result of caspase activity within a dying tumor cell. So prostaglandin E2 can have some pro-proliferative and immune silencing effect, both of which are very harmful for a cell which is neoplastic or cancerous in nature. So it is leading to proliferation of cancerous cells or the surrounding cancerous cells. That is, there is the proliferation of cancer stem cells in particular. And additionally, it is also creating an immunosuppressive environment, or it is silencing the immune activity within that region, because it is not allowing the entry of immune cells in order to mediate clearance. So while one particular tumor cell is undergoing apoptosis and getting cleared off, it is allowing or supporting the survival of surrounding cells, particularly of stem cells, and is also creating an immunosuppressive environment which will protect those cells from the action of the immune system. So this becomes a pro-oncogenic effect. Then in the second panel, which is panel B, we are seeing that apoptosis of tumor cells that are treatment responsive can provide a vacant niche into which more competitive tumor cells can proliferate, ultimately leading to tumor regrowth and resistance. So what it does is, if you have read about the concept of selection pressure, it basically means that you will have um, a condition like, if you impose some sort of a stress on cell because of the nature of proliferation of these cells, where they undergo rapid proliferation, rapid incorrect random proliferation, they incorporate a large number of mutations during the process of dividing. Now when they're dividing, it might so happen that the kind of mutation that has been incorporated within one particular cell is actually pro survival. For example, say if we are treating cells with one particular drug, say cisplatin, for example, while it is not a very specific example, say any other drug, any particular drug which is um, meant to be a chemotherapeutic treatment, it might so happen that over the course of divisions that population of cancer cell has undergone, one or two of those cells might have accumulated or might have incorporated a mutation that is allowing them to survive in that 5FU or cisplatin containing environment. When it does so, that clone gets selected for. So under the pressure of the drug, under the selection pressure of the drug, only that particular clone which is pro survival, or which can be considered, according to Darwin's words, the survival of the fittest, or the most appropriate survivor, will be selected for, and only then that competitive clone will keep on dividing, because now it has found a condition which is beneficial for it instead of being harmful. So under the presence of a selection pressure, in a large number of cases, it is possible that the treatment responsive cells, while they die, they are creating that immune silencing environment, and they are supporting the proliferation of these highly competitive clones and allowing them to survive further, which will lead to tumor regrowth and ultimately also lead to therapeutic resistance. Thirdly, apoptotic cells also release a number of eat me signals and find me molecules such as FKN, which is fractalkine, ATP, and lactoferrin to signal their removal by phagocytes. So eat me signals are responsible for calling clearing cells, or calling sentinels which will come and remove them from that environment, and that is a desirable phenotype under normal circumstances. However, it can be such that these signals will be pro-tumorigenic in nature, and many a time they can cause the polarization of macrophages. So they can turn the tumor-associated macrophages, or TAMs, as is mentioned here, towards a pro-oncogenic state. To be more precise in this regard, they can stimulate angiogenesis, tumor cell motility, and cancer cell dissemination. And they can prepare the pre-metastatic niche. They can silence immune cells. They can prevent NK cells and T cells from attacking the cancer cells. So what the ways in which it is doing this is that, first of all, it is polarizing the macrophages, or more particularly polarizing the monocytes into M2 class of macrophages. M2 are usually the pro-tumorigenic macrophages, which um, help or which allow tumor cells to not be detected by the immune system, and therefore it is considered to be a tumor immune escape. Secondly, they can also, via ATP and LTF, lactotransferrin, they can proliferate further, and it will help them in tropism. Tropism basically means that for a cancer cell, or for from a population of cells within a tumor, if a few cells are sloughing off, or they are getting detached, they are traveling through blood and through the circular system into eventually circulatory system into eventually a new site, then that particular region also needs to have a very favorable environment. And the tendency of cancer cells to go and colonize that favorable site is what is understood as tropism. So different types of cancers have different tropism. Breast cancer often can be seen to have some brain tropism. For prostate cancer, we see that in case of brain, in lungs, liver, bone marrow, you see maximum tropism. So depending upon the type of cancers, the nature of cancer, the type of mutation those cancer cells are carrying, the localization to different sites can be different, and that is known as tropism. And additionally, apoptotic cells can also lead to pro-angiogenic proliferation. Angiogenic proliferation basically means that there can be more and more formation of blood vessels, more and more enrichment of vascularization at a particular region, which will allow tumor cells to survive. So it is creating more and more blood flow for tumor in a particular site, and it is also anti-apoptotic in nature. Therefore, it is supporting the growth of neoplasms instead of opposing it. So that is what we see here. Now here you see a sort of comprehensive view of what exactly happens in context of cancer, what exactly happens with apoptosis in context of cancer. So death receptors like FAS and trail R that we just talked about can have non-apoptotic signaling functions. What type of functions? Either it can promote the growth, invasion, and survival of cells. All of these will support development of cancer and progression. So death receptors can also engage the CAD, which is caspase activated DNAs, uh, and that leads to DNA damage and mutagenesis. The intrinsic apoptotic stimuli can engage limited mitochondrial outer membrane permeabilization or minority MOM, which activates effector caspases to sublethal levels. So what it is effectively doing is that when under certain circumstances, when apoptosis is not being activated to the threshold that it needs to be activated, there is a sublethal response which is occurring inside the cell. That there is activation of caspases, but it is sublethal in nature. There is permeabilization of the mitochondria, but that is also sublethal in nature. So what it effectively does is the endonucleases, which are CAD and endonuclease G, which are released after there is permeabilization of the mitochondrial outer membrane, they are going and cleaving double-stranded DNA. Now they are cleaving in a very non-specific manner or in a very random manner, or they are creating DNA damage. They are breaking DNA at random places because that cell is tumorigenic, and because that cell is already carrying mutations, and it has a improperly functioning repair system. What will happen in such cells is that there will be random ligation of different regions, random incorporation of bases, so that will lead to more and more oncogenic mutations, creating more deadly cells instead of leading to appropriate apoptosis of cells that were supposed to die. So in this case, the improper activation of apoptosis or the um, sublethal activation of apoptosis can have functions that promote the growth of cancer or the development of cancer, and in fact also support its progression. So what we are seeing is that I think I've said this before also, that when everything is working right within a system, you are you are a healthy individual. Any of these basic systems, any of these basic protective systems that we take for so easily take for granted within any living organism, when any of these systems go awry, it leads to these sort of conditions, and most commonly leads to cancer. Whenever there is an effect on the immune system, on the cell division, on cell cycle, on cell proliferation, all of that eventually leads to neoplastic transformation or cancer of the cells that we are talking about. So that kind of sums up the idea of how apoptosis is affecting cancer. Now, because it does so, it can also be looked at from a therapeutic point of view, that if indeed there is so much of an involvement of apoptosis and cancer progression, then what can we do in order to manage cancer, or in order to facilitate new approaches of chemotherapy? So what you see here are different classes or different sub-steps at which the regulation of apoptosis is ultimately um, messing up the checkpoints that are normally present in cells and eventually leading to cancer. And how at each step you have some inhibitor or you have some therapeutic strategy which can control the same. So here in this particular schematic, what we see in pink are the enhancers are apoptosis, and the inhibitors of unwanted effects are shown in these blue boxes, and we have some examples for each case as well. And some of them are even approved in clinical trials. So given that they do not prevent death following the widespread apoptotic mitochondrial outer membrane permeabilization or MOM, caspase inhibitors could prevent the unwanted effects of caspase activity. That includes DNA damage and prostaglandin E2 production. So first we see that BH3 mimetics like venetoclax and navitoclax are present, which are acting at the which are mimicking BH3, and they are controlling mitochondrial outer membrane permeabilization itself. Then we have SMAC mimetics, we have caspase inhibitors, caspase activators, CSF1R antagonists, and PGE2 inhibitors. The inhibitors of PGE2 activities, such as celecoxib, could be applied in combination with apoptosis-inducing cancer therapies. So it is basically an approach of combinatorial therapy where one group of therapies are inducing apoptosis, promoting cell death, but at the same time, you're also giving an inhibitor of PGE2 so that upon cell death, whatever prostaglandin is being produced is not able to execute its unwanted or pro-oncogenic activity, and therefore inhibitors such as celecoxib are being considered under these circumstances. So therefore these two can be used in combination. Similarly, inhibiting CSF1 or colony stimulating factor 1, which is responsible for the polarization of monocytes into macrophages, that can also be stopped. Either you deplete those tumor associated macrophages or M2 macrophages, or you can switch them to the anti-tumorigenic phenotype, which is M1. Following failed apoptosis, the enhancement of caspase activity using SMAC mimetics, which will antagonize XIAP or other means, may also promote apoptosis. That is yet another approach. Likewise, direct caspase activating molecules may also preferentially promote tumor cell death that have undergone failed apoptosis and that have a low level of caspase activity. So these are the different approaches, and what you see next to each of these drugs are the different commercial names or the official names of these drugs and what stage of clinical trials they are in. You will see that some of them are FDA approved, for example, venetoclax. We have some which have completed 4 levels of these trials, for example, celecoxib, and after all the levels of clinical trials are carried out, then they are marketed or acceptable for marketing or for commercialization and for patient treatment to be actually carried out. So it's a long process, but certainly it is necessary because these are very, very serious drugs, and the way in which they work has to be taken very, very seriously. So with that in mind, I'll just give a brief overview of the process of cancer, the substeps of cancer. So first of all, the definition of cancer. While you'll find a very large number of definitions which each point in different directions, the basic idea of cancer, particularly according to the American Cancer Society, is that it is a group of diseases which are characterized by uncontrolled growth and spread of abnormal cells. So each of these terminologies are important. First of all, it's not a single disease, it is a group of diseases. Why do we say that? That is because a glioblastoma and an osteosarcoma are completely different. This is brain cancer, this is bone cancer of certain types. Their manifestation is very different. The way in which they are detected, their cause, their effect, their survival, their treatment approaches, they are different. So each of these are handled in different manner. They are diagnosed and their prognosis is carried out in different manners, but effectively they are cancer. So cancer is considered to be an umbrella term under which you have a group of diseases. What is the key feature by which it is characterized? There is uncontrolled growth of cells. That is, cells which are not supposed to grow are growing and growing uncontrollably. And they are spreading to different sites, which is what is understood by the process of metastasis. That cells which were supposed to be in one tissue within one organ of the body are spreading anywhere and everywhere inside the system, and these cells are not normal. They are abnormal cells which are carrying some sort of mutation, some errors, which is what has given them this property of spreading randomly to any and every region. And therefore, the definition is composed in this manner, that it is a group of diseases characterized by uncontrolled growth and spread of abnormal cells. So with this in mind, what we see here are the basic stages in cancer. You have initiation, hyperplasia, dysplasia, in situ cancer, invasive cancer, and metastasis. The definitions of this are something I'll come to later as well. But if you see it characteristically, what you see is that within the epithelium, this is again a generic definition. It will depend or it will vary based on the type of cancer that you're talking about. But the basic idea is that within the normal epithelium, we are talking about epithelial cancers because they are most commonly seen. There is some sort of an initiating event. Here, the example given is, say, the activation of an oncogene in the in one particular cell, which is undergoing that alteration. There can be n number of other factors, but there has to be some initiating event which has occurred. Some carcinogen, some mutation, something has come in. When it does so, there can be the random or rapid division of this particular cell, which is dividing at a much higher rate than a normal cell. This condition or this situation is known as hyperplasia. Then comes dysplasia. So cells that have altered and that carry this increased growth potential are being considered under dysplasia. I'll give a little more detail into the definition of dysplasia in one of the upcoming slides. But for now, just take this into consideration and keep this image in mind where you can see that the initial cell, which was orange in color, initially altered cell orange in color, different from the normal epithelium that is yellow in color, now has a subpopulation which is even more different. You are seeing a purplish color that has come in. This is important. It will come later. Then we have in situ cancer or in situ carcinoma. In situ literally means at that inside or at that site. So within the site where the event had initially occurred, only within that site you are seeing that there is the formation of a cancer or a carcinoma or a huge mass of cells or a tumor is formed. The purpose of saying that it is in situ cancer is that it means that the cells in this region are intact and they are localized in that place. They have not breached the basement membrane or they have not breached the epithelium and they have not invaded within the tissue or within the circulatory system and they have not gone anywhere else. So this is in situ cancer. The next step is invasive cancer. In invasion, we see that there are a number of sub steps within that green box. You'll be able to see the first step is invasion, like I said, that they are breaking or breaching the basement membrane and they are invading. Once invasion has occurred, the next step is intravasation. Intravasation literally means entry into the vascular system. So they are entering into the vascular system via entry through the endothelial cells which line the vascular system, the blood or lymph, whatever it is. Mostly they travel by blood. They can also travel by lymph and then localize to lymph nodes, which are also a very, very common site of metastasis of many cancers, particularly of epithelial cancers. So first step is invasion, where they are breaching the basement membrane. Second step is intravasation, entry into the vascular system. Third step is dissemination. Dissemination means they are separating or they are moving about or disconnecting from the original tissue to other locations. So they are traveling through blood and they are disseminating to some other site. Once they have done so, at the site which is conducive for their growth, which is fertile for their growth, or where they are capable of which is a good site for tropism, where they have a tendency to go. Once it reaches that site, there is extravasation, which means exit from the vascular system, and colonization, which means now it will colonize in a new site and it will form a new tumor and it will form a new secondary tumor, which is otherwise known as metastases. And this process of moving from the original location to a new site via the circulatory system is known as metastasis. So there are primary tumors, there are secondary metastases, among which you have micro and macro metastasis depending upon the site or the location and the size of the tumor. And the entire process is known as metastasis. So that is the spread of abnormal cells which has occurred after uncontrolled growth. So I hope it was clear. It was quite a lot of detail, but these are the sub steps that basically occur. Now, in three very landmark articles, two scientists known as Douglas Hanahan and Weinberg, Hanahan and Weinberg, they both have written a series of articles where they have defined what are the hallmarks of cancer. And they have also added additional hallmarks and enabling characteristics to these. You don't need to get into detail, but these are the basic ideas that they have given. So what effectively it means is that there are some uh, common hallmarks or common characteristic features of any cancer cells. So that includes, of course, sustained proliferating signaling, that there has to be proliferative signaling. The cells must grow in number. Second, they must evade growth suppressors, which means that anything which is preventing them from growing, that has to be evaded or has to be avoided by cancer cells. Third, they have to avoid immune destruction, that they have to create an immunosuppressive environment so that the immune system does not come and kill them and clear the area of the tumor. Fourth is enabling replicative immortality, which means that cells that are undergoing cancerous cells or cells that have undergone the neoplastic transformation should not die after a certain number of divisions. They should be capable of constantly dividing, or they must attain replicative immortality. They should not die after a certain number of passages. And of course, one of the ways to do that is by overexpressing telomerase that we have previously talked about, will keep on which will keep the chromosome ends intact and will allow these cells to keep on proliferating randomly. And then we have tumor promoting inflammation. So it will push for such changes, or it will push for such inflammation which allows the growth of tumors. It will activate invasion and metastasis that we just discussed. It must access the vasculature. It must promote angiogenesis. It must promote the formation of blood vessels so that that region can be enriched with whatever nutrients and oxygen is required for the sustenance of such tumors. It must the genome of such cells are not very stable. They must incorporate mutations, which allows them to adapt very rapidly to whatever changing environment they are in. And finally, they must resist cell death, which means they should be able to evade cell death, or the effective survival of the tumor must be prolonged, and therefore pro-apoptotic signals should ideally be ignored and anti-apoptotic signals should be shot up. And cellular metabolism also needs to be altered. One common example of that is the Warburg effect, which you have also already studied in the lecture. So it leads to additional excessive glycolysis, which allows for the cells to proliferate very, very, very randomly. So that is the way in which these steps are carried out. And you also have some emerging hallmarks and enabling characteristics like senescent cells, polymorphic microbiomes, non-mutational epigenetic reprogramming, and phenotypic plasticity. These are these are more advanced concepts. We don't need to get into them right now, but basically what you need to understand, there are some defined hallmarks. Again, this is something which is constantly changing. The first hallmarks of cancer, like you can see in the article here, were defined in the year 2000. Then there was the next generation of hallmarks where new hallmarks were included. Then the latest publication was in the year of 2022. Again, we can expect another 10 years from now, we'll have more updates on it. And this evolves with more and more knowledge, with more and more treatment, with more advancement in technology, and we'll be able to find out more details about the same. So, keeping this in mind, we will look at each of these substages of cancer that are normally studied and studied histologically. In fact, that you take patient biopsies and you section them, you stain them, and you see what is the condition of those stage, which stage that particular patient is in, how far cancer has spread at that point of time, and how that patient needs to be treated in under such circumstances. So there is a progressive development or a progressive um, neoplastic transformation of cells that occurs. So between the extremes of a normal tissue and a very highly malignant tissue, there are a number of substeps or a number of morphologies that you can visualize under the microscope. So you will have some growths that have cells that deviate only very minimally from normal tissues, but they might be abnormal in that they have excessive number of cells. So only when the cell number has increased, such a growth, like I previously said as well, is hyperplasia. So you can see that in the histological section as well, that you have a group of cells which are very darkly stained because of a number of nuclei that are present, and that is because of hyperplasia or excessive proliferation that has taken place. They still retain the ability to assemble into tissues that appear reasonably normal. So hyperplasia is not always considered to be a as a very serious condition, or not necessarily considered to be a very cancerous trait. It is often considered even to be a precancerous lesion. Second, we have metaplasia, which was not there in the diagram, but what it effectively is the subsequent step after hyperplasia. We have metaplasia. In metaplasia, one type of normal cell layer is displaced by cells of another type that are not normally encountered at this site within this particular tissue. So in metaplasia, you have in a normal tissue, you have one particular type of cells which are present in that region. When this particular group or this particular layer of cells is getting displaced by another type, which is abnormal in nature and which is not supposed to be present at that site, these invaders, while they are normal in nature, they can, while they're visibly normal, they are not supposed to be at that location. That is metaplasia, which is another uh, sub-step that is often observed during the progressive development of tumors. One of the most common examples of this is the presence of Barrett's esophagus, under which circumstances we can see that there is a metaplastic Barrett's epithelium. So one type of cell which is like within the esophagus, you are able to see cells from a different region. So this is metaplasia, that while the cells will look normal, they are present at a site where they are normally not supposed to be present. So that is metaplasia. The third type is dysplasia, which is used to refer to an abnormality of development. And if you remember the diagram as well, we saw that within the tumor, there are cells in the center which are different morphologically, and also like it was denoted in terms of color. But basically, these are cells that have undergone some sort of a change, and they are visibly very different. And in other words, they are cytologically very different, and therefore they look different under the microscope. So cells within a dysplasia are cytologically abnormal, and you can see that under the microscope as well. When we say that it is cytologically different, it basically means that there is a change in the type of cells, or the nuclear size, the nuclear shape, the nucleus versus cytoplasmic ratio, more mitosis, etc. So when we say nuclear versus cytoplasmic ratio, it means that within a cell, the percentage of area that is occupied by the cytoplasm and the area that is occupied by a nucleus has shifted. So for the same size of the cell compared to a normal cell and a cancerous cell, you will see that the size of the nucleus is much bigger. That is because DNA replication is occurring randomly. Such cells are dividing very randomly and dividing at a very fast rate. So under such circumstances, you are able to see that there is a shift in the nucleus versus cytoplasmic ratio. And in fact, you can see this in the histological section on the right as well, that it is a dysplasic epithelium of the cervix. And in the upper layer of cells, you can see that there are very large nuclei which are darkly stained, versus the basement membrane, which still has normal cells. It has much smaller nuclei. So the nucleus size, the nucleus shape, the nuclear cytoplasmic ratio, all of that has been altered. And therefore, seeing it under this section, you will be able to distinguish what state it is currently in. So that was dysplasia. Then we have anaplasia. Anaplasia refers to a change in the structure and cell orientation. You will find that an anaplasia is a condition where cells have um, sort of reverted to a more primitive phenotype, which means there is instead of going into a more differentiated state, they are going towards a more uh, proliferative state. Let me reframe this a little bit. So basically, when a cell is dividing, after it has divided, it has two possibilities. So within a tissue, you have mostly a population of cells which are differentiated cells. They have attained their proper function and they are capable of executing whatever functions are required for that tissue to function properly. But within that tissue, there are some cells which are known as the stem cells, which are not differentiated, and instead they have the potential to divide and to generate differentiated cells. So within a tissue, you do not expect cells to have this de-differentiated or undifferentiated sort of phenotype. No, they are supposed to be differentiated. But when a particular individual has the cancer of that particular tissue, you will see that these cells eventually go into a state where they stop preferring differentiation and instead prefer to go into division. And therefore, you will see this shift in the balance from differentiation towards division, so that the cells can proliferate randomly and they can create large masses of large tumor masses within different locations within the body. And anaplasia is considered to be a characteristic of malignancy, that when you are seeing that within this particular tissue, what, for example, what you see in this sort of pink H&E stain section here, you can see that these cells do not have a very distinct or very well defined morphology. No, they have more of a rounded, undifferentiated sort of structure, and it means that the cancer has now progressed to a stage where it is most definitely going to invade very soon, and it is going to be a very serious case, and it is considered to be malignant in nature. And finally, we have neoplasia. Neoplasia refers to the process of abnormal and uncontrolled growth of cells. And those cells are known as neoplastic cells. The growth of the cell is excessive. It is uncoordinated with the normal tissues that are surrounding it. And the product of the process of neoplasia is a neoplasm, which is a tumor. A neoplasia can be in either of these stages. It can either be benign, which means that it has not yet moved to any other regions. It is just a simple surgical removal. You will often find people having, little masses in random places within their body, which are just surgically removed, and they do not need to undergo very extensive chemotherapy or immunotherapy or any of those very critical and often life taking sort of treatments. They're very, very severe treatments. So these are more easy to treat. They are surgically removable, and they are not very, the life risk associated with them is much lesser. So that those are benign tumors or benign neoplasms. You have potentially malignant neoplasms, or you have malignant neoplasms, which are cancerous in nature. So what this effectively shows is that not every tumor is a cancer. Sometimes tumors that many often, in many cases, you can find that women who towards the age of 40 or in the later stages or close to their menopause, in a large number of cases, they develop ovarian tumors, ovarian cysts, which we commonly know of them as, and they can be removed by laparoscopic surgeries or basic surgeries. It's a relatively easier approach to take, and it is not very life threatening for such individuals. So in such cases, they are said to be more benign. They can be removed easily, and such patients do not have to undergo very serious treatments. On the other hand, if a particular individual is seen to have these abnormal cells, and they have seen to be carrying anaplastic neoplastic cells, or they have a mixture of these cells, then you can most definitely be certain that those neoplastic cells will take over. They will not allow the normal cells around them to survive, and they will proliferate uncontrollably, and they will form masses of cells, and they will form tumors, and eventually that will lead to such a person will be diagnosed with cancer, and in the worst case scenario, that cancer will metastasize, go to other organs, and can compromise the individual's life to a huge extent. So what this slide basically was meant to show you was that there are multiple stages. It's not always very directly black and white, that every outgrowth you see or every um, a random growth that you see within the body is not cancer, versus every simple mole that you can observe in your body is not safe. It can be cancerous, it can be indicative of melanoma. It is context dependent. And therefore, what is required is that you get it checked, you get it biopsied, and you get it checked, and you make sure that in case, in the worst case scenario, if that is a cancerous lesion, then it gets treated, it gets diagnosed and treated at the correct time, and it increases the chances of survival of such patients. So, yeah, this is the final slide that I'll be showing for today, and then we'll discuss some sample questions. Um, there are two potential models that are usually looked at with respect to cancer and how cancer is spreading in the long run. The first is the clonal evolution model. The second is the cancer stem cell model. In the clonal evolution model, what you can see is that from one particular cell, one particular clone, basically, you see that there are some cells which are from one cell it is dividing into two, from two to four, and so on and so forth. So each clone is dividing and it is forming a certain mass of cells. From these clones, some of them, they incur some sort of a mutation or some sort of a change, and they become different, or they acquire different properties compared to the parent cell, which is indicated here in blue. And those cells will then proliferate, and those might eventually be selected for. So the concept of it is that from one clone, you are seeing that in subsequent rounds of division, some clones are acquiring some mutations and they are getting selected for in the long run. The second is the cancer stem cell model, in which we are saying that from the entire tumor, there is one particular cancer stem cell which is responsible for producing more and more clones, or producing different clones compared to the parent cell, and not the others. So there are two approaches. Both of these have been supported and have been debated against. There are theories which work in either direction. But yeah, you cannot have one hard and fast rule. Till date, there has not been a very hard and fast rule as to how cancer cells are selected for, and how only one branch of cells are being accepted or the other branch is not accepted, or what exactly is happening inside a tumor. So these are two approaches that have been seen, either that only some cells are getting selected for. The cancer stem cell model says that only the stem cells are selecting for which cells will proliferate in the upcoming generation. So there are two lines of thought, or two schools of thought, which are currently being explored. And finally, in this rather confusing schematic that you see on the right, this is the representation which was shown in the second hallmarks paper by Hanahan and Wigberg. So it is basically a representation. It's not the actual network which is persisting inside the cell, which is even more complicated, but it is just a representation of how different types of circuits are all coordinating with each other in order to confer the hallmark properties that we talked about to a cancer cell. So we have motility circuits. Motility circuits include, when we say motility circuit, it basically means circuits or signaling pathways that are allowing these cells to become motile, or for them to move or metastasize from one region to another. The second are cytostasis and differentiation circuits, which are controlled by some anti-growth factors, which are now no longer functional. So those set of cells are there. We have viability circuits, which are allowing cells to be viable, or allowing cells to persist despite the pro-apoptotic signals that might be there. And of course, we have proliferation circuits as well. So each of these circuits are very broadly classified, under which you are having multiple mediators. Like, for example, in proliferation circuits, we have growth factors, which are working through, say, receptor tyrosine kinase. One example would be EGF, epidermal growth factor, that works through receptor tyrosine kinases via the RAS oncogene and mediates the expression of MYC and many other genes which will confer hallmark properties. You have survival factors, you have cytokines, you have death factors which are no longer functioning here, or even if they function, they're working in a pro-oncogenic manner. You have proteases which are digesting the integrins that are normally responsible for holding the tissue together, and those are being disrupted so that the tissue can pinch off from its original site and can move to some other location. So all of these different mechanisms are in place. They are operating in a crosstalk manner, and it's not always necessary that all these mutations have to occur inside a cell for it to become cancerous. No, only a few, a subset, or at times even one or two mutations are good enough for a cell. If they are that critical, if they are affecting such crucial genes inside a cell, then that can lead to very far reaching consequences. The underlying thought here is that if an initial mutation has caused a cell to become to activate an oncogene or to become neoplastic in nature, then it will keep on dividing and it will keep on incorporating more and more and more mutations in each generation, and that will lead to a complex mutation which will eventually make it very difficult for us to identify that what was the first mutation that caused it, what was the driver mutation. These are terminologies used in molecular oncology, that the initial mutation which has caused the cell to become cancerous is a driver mutation, and everything that it has acquired over time is a passenger mutation. So it's a little difficult to identify that. There are technologies which are being put in place to help identify that so that we can possibly find some therapeutic application for the same. But the effective thought that I want to get across to you is that there is a huge circuitry that is in place, that is making sure that each of your cells are healthy, functional. In case not healthy, they should be eliminated. In case they are not healthy, we can try to repair them, and if the repair machinery is also tired and exhausted and not able to repair it, then that cell should die. So when all of these mechanisms are just not functioning properly, only then do people suffer from cancer. And despite all of this being in place, along with an immune system that is doing its job beautifully under normal circumstances, when all of these things go wrong, along with lifestyle habits such as smoking, such as exposure to radiation, etc., all of that together is what can lead to cancer in individuals. So this is a disease that is very difficult to understand. And the more you understand, the more questions it brings you than answers. But yes, it is certainly a fascinating topic. If it is something that interests you, you should go ahead and look into it as well. So with this concluding line, I will now switch to some questions. If you have any questions, please go ahead. I'll just open this. I hope it is visible. Yeah, okay. Visible. Okay. So, this was one of the previous year assignments. That was about the first question is about the correct statement for senescence. So senescence cells, they remain irreversibly arrested at G0 phase. It is an active cellular death process. It occurs only due to infection. Chromosome fragmentation and cell shrinkage are the main characteristics, or all of the above. So among these, the correct option is that senescent cells are arrested at the G0 phase. Senescence itself is a form of cell cycle arrest, and that arrest occurs at the G0 phase. The remaining options are not true. It's not a death process, cellular death process. We discussed that those are apoptosis and necrosis. Does not occur due to infection. It is a normal physiological process that the cell will go into when it has some external stimulating factors. Chromosome fragmentation and shrinkage are also not a part of senescence. So the correct option is only option 1. The second question is about which of the following is not a hallmark for aging. So we discussed that different types of hallmarks that we saw. So you have telomere attrition, that if the telomere ends are getting pinched off or eroded, then that is considered to be a hallmark for senescence and for aging as well. Mitochondrial dysfunction was also a part of it. Stem cell exhaustion was also a part of it, but low ROS or low reactive oxygen species are not usually considered to be a hallmark for aging. In fact, the excessive production of ROS is not beneficial, and that can lead to random breakage within the genome. So the ratio or the levels of reactive oxygen and reactive nitrogen species within the physiological system are also very critical. And if there is significant fluctuation in the same, that can generate DNA breaks, single-stranded DNA breaks, and that can lead to that can incur mutations, that can lead to random joining, or all these different sorts of mutations, and that can be a critical situation for the cell as well. There are mechanisms also in place for it. There are innate immune responses that work in that direction. There are cellular compartments, particularly peroxisomes, where this is also taken care of. So there are operating mechanisms that have been put in place to take care of reactive oxygen species. But yes, it is certainly something that needs to be taken into consideration. Um, which of the following diseases is a consequence of too little apoptosis? So it was there in one of the lectures as well, that apoptosis has a role in different disorders. You will find review papers on this as well if it is interesting to you. But when there is very little apoptosis, that actually leads to autoimmune disorders. But yeah, let us go through the options anyway. We have autoimmune diseases, Parkinson's disease, AIDS, and stroke. So among these autoimmune diseases have actually been seen that when sublethal or very minimal apoptosis occurs, then that can trigger autoimmunity in patients. Um, then in question 4, we are asked which of the following proteins can physically go to the mitochondria to promote the release of cytochrome C. So we have BAX and BAC, we have BCLXL, we have BAC FAF1 and BCL2. So if you remember that schematic representation, so the main proteins that are operating to execute MOMP, or the permeabilization of the mitochondrial outer membrane, are BAX and BAC. They form this complex and they go and bind to the mitochondria. They form channels in the mitochondria, which disrupts the mitochondrial outer membrane, permeabilizes it, and releases cytochrome C into the cytosol, which triggers the extrinsic pathway of apoptosis. So it is BAX and BAC. And which among the following proteins inhibits apoptosis? So we had a list of factors about pro and anti-apoptotic proteins. So among them, BCL2 is a apoptosis inhibitor. So that would be the correct option in this case. Um, which of the following is a proto-oncogene? So I don't think I mentioned it directly, but yeah, there are majorly two sets of genes. We have proto-oncogenes and we have oncogenes and we have tumor suppressors. So oncogenes under physiological conditions are present as proto-oncogenes, which when mutated or when altered or when activated by some external factor can be converted into oncogenes. Some examples of these are, of course, the correct answer to this, which is c-Myc. We have RAS oncogene. So these have the potential to be altered and to function as the trigger or the driver mutation, which can convert a cell to the cancerous phenotype. On the other hand, there are other set of proteins inside the cell or other set of genes inside the cell, which act as sort of security guards inside the cell. They maintain the correct functioning and homeostasis and health of the cell. Those are known as tumor suppressors, which under normal circumstances will prevent the formation of the tumor. And when there is the suppression of, or when there is the mutation or alteration of tumor suppressors, then the cell will become cancerous. So PRB and P53 and BRCA1, in fact, PRB and P53 are tumor suppressors. BRCA1 has a different role. BRCA1 has a role in DNA repair, and therefore it acts as a sensor of DNA damage repair and brings the repair machinery to identify that error and to correct it. So among these, the proto-oncogene is only c-Myc, and it was in fact even present in the circuitry schematic that I showed you. Um, in C. elegans, which of the following active proteins would prevent apoptosis? SED4, SED3, SED9, or all of the above? So if you went through the lectures, you would see that there was a comparison drawn between C. elegans and the human or mammalian genes that we are familiar with and how they can be considered to be analogous with respect to caspases. So among them, the inhibitor or the preventer of apoptosis was SED9. So again, a very direct question. So if you just have to go through the lecture notes carefully. So SED9 is responsible for the prevention of apoptosis. Pick the correct statement about cancer cells. So yes, here. Okay. Pick the correct statement about cancer cells, abnormal unrestrained proliferation, invasion of surrounding normal tissue, cancer cells of somatic mutations, or all of the above. So, of course, all of these statements are correct. We have seen that there is abnormal proliferation or hyperplastic growth. There is invasion or a breaching of the basement membrane, which is a part of metastasis, and they incur a lot of somatic mutations, which is also considered to be an initiating event. So all of these statements would be correct in that case. Uh, then the next question is about caspase 3. So, which of these statements are correct about caspase 3? First, that it causes DNA fragmentation by activating DNases. Um, yes, it does. So, caspase 3 is one of the effector caspases. So, it causes fragmentation of DNA during apoptosis, which is a pre-lytic event that occurs during the process of cell death, programmed cell death. Second, it dephosphorylates the myosin regulatory light chain. No, it does not do so. It is activated by ROC1 kinase. Yes, that is correct. I don't think it was part of the lecture, part of my presentation, but it was there in the lecture that ROC kinases, they are activated by this particular caspase 3, and therefore, in a lot of in certain assays, when we are culturing cell, we use ROC inhibitors in order to prevent cell death or cell apoptosis, and therefore it is significant in that aspect. And activated caspase 3 leads to membrane blebbing. Yes, that is also true because that is also one of the characteristic features of the entire process of apoptosis, or in fact, one of the key morphological features that are observed in a cell that is undergoing apoptosis. So statements 1, 3, and 4 are correct. So yeah, that would be option 2, and therefore that is the correct option. Yeah, I think it is closed. Okay, so with this, I've come to the end of today's class. So to sum it up, what we basically looked at initially were the phenomena of aging, senescence. What is the significance of senescence? What are the hallmarks of it? What are the hallmarks of aging? Why it is needed? What is mediating it? What are the key processes that are triggering senescence? What are the other processes? This senescence is not a cell death process, but what are the other processes which the cell can enter into it if it is deprived of nutrients? And the key difference with senescence was that a cell enters senescence despite the presence of adequate nutrients and adequate survival signals in its surrounding environment, whereas in the other case, this is not true. And among the cell death processes, we looked at apoptosis and necrosis. What are the differences between them? How they are regulated, how they are differentially regulated, what is the need for each, and how they are executed under normal and under disease conditions in individuals. And we also saw the role of apoptosis in cancer and how it can promote cancer under certain circumstances. And we understood some basic properties of cancer, some basic steps and sub-steps. The signaling pathways are another extensive domain of study. I did not get into it because it would not be possible to cover it in this class by itself. But yeah, it was there in the lectures, and it is also something which you'll get hundreds and thousands of reviews online if it is something that interests you. And it will help you appreciate how all of these pathways are all connected at the end of the day, and how crosstalk between these pathways is so crucial. And finally, we looked at some of the key stages and the key histological sections that can be observed when a particular pathologist is looking at a biopsy, and what are the differences between them and the way in which cancer cells evolve so that they can move from the primary site of tumor formation to a different metastatic site and can colonize there. And we looked at some questions for the past week and for this week, and keeping all of that in mind, I think I am done for today's class. If there's any question, please go ahead and ask, and then we can wrap up for today.
Thank you, ma'am.
Do you have any questions?
Ma'am, I have small doubt.
Yeah.
But these assignment questions are repeated in final exam, ma'am.
That is very difficult to say. You might get some questions, but not necessarily exactly the same. You can look at them for reference. You might get similar questions, But in your exam, you will probably get a different set of questions, not exactly from the assignment. They are just meant to prepare you and they add up to your final grade. Any other questions?
Ma'am, these live sections also used for ma'am.
Sorry?
These live sections also used to final exam, ma'am.
Um, I don't know. That depends. So they are recorded and they are kept. They are available with the team. So it depends upon the team and the examiner, whoever is setting up the question. It's not directly in our control. From your end, you can go through the topics and you can look at anything, any area which is not clear to you, and you can bring it up. We'll have an extra session on 21st, which is a Tuesday, I think, to clear up any doubt throughout the course. And that will be another 2 hour session. So, any doubts from all of the 8 weeks, we'll try to address those. And any extra information, any extra questions that I can find, I will also incorporate those. So, that is the purpose of live interactions. And if you have any other doubts that you want to ask the course instructor directly, Professor Lalorea, then you can ask it on the discussion forum, and she would probably help you get through with that.
Okay, thank you.
Okay, thank you so much for joining, and I'll see you for the next session on next Sunday. So, thank you so much, and good night.
Okay, ma'am.